Biology Part 210. We've been talking about Gregor Mendel for our understanding of the inheritance in plants. All the seeds produced called hybrid seeds were collected and used to grow new plants in subsequent years. In one particular experiment summarized in the figure, Mendel crossed and hybridized two pure breeding varieties, one with the smooth rounded seeds and the other with the wrinkled seeds. In the next generation, they were called the offsprings. Well, number one, there were generations. All the seeds were smooth, irrespective of whether they developed on a parent plant belonging to the wrinkled strain fertilized with the pollen from the smooth strain of the vice versa. Mendel collected 253 of the hybrid seeds and grew them into the new plants, where he allowed the flower and set seed by self-fertilization in the normal way. The new seeds were the grandchildren of the original cross called offspring. They were number two generations. There were 7,324 seeds, for which 5,474, almost exactly 3/4 were smooth, and 1,850 were wrinkled. From the whole range of the similar results, Mendel reached an important conclusion, which is now known as the Mendel's first law, the law of segregation, or less commonly known as the law of particular inheritance. This states that an organism's characteristics are controlled by the factors which are normally carried in pairs, but which occur singly in the gametes. It is now known that this occurs because the cells of the most sexually reproducing organisms are diploid. That is, they carry two sets of homologous chromosomes, whereas the gamete cells are haploid and carry only one. The character which appeared in the offspring are called the generation 1 smooth seeds. The examples above of the Mendel called the dominant, and with the character of the temporarily obscured wrinkled seed was called the recessive seeds. Mendel's factors were purely hypothetical and their physical form remained understood for 100 years. The modern equivalents are called the genes, which is the short length of the DNA carrying decoded instructions for the development of RNA, representing the factor of the smooth seeds as S and the factor for the wrinkled seed as S in the lowercase. S in the uppercase is for the smooth seed. Mendel's experience can be explained in the dominant smoothly factor of the recessive wrinkled. These factors are called the allelophores or alleles, alternative forms of the gene for one characteristics, or the phenotype of the male parent smooth seed. Well, all the alleles are carried in the pairs. Since it is the pure breeding line, the corresponding genotype is SS. This homozygous dominant genotype is the genotype of the pure breeding wrinkled. Female is homozygous recessive as SS. The gametes formed in each pair of the pollen grains contain the alleles of S. Then we've got the Punnett scale method as illustrated. The list of male gametes is transferred to one side of the table, while the list of the female gamete is copied at the top. For this cross, the table consists of 4 boxes which are now filled. Considering the first entry, you can see that when two gametes are there to fuse, their chance and the probability is 1 out of 2, that is 50%. the male gamete will carry the S allele and a male gamete will carry it or may not carry it.